Done, sir.
May I
Himani,
start, sir?
Himani, go ahead.
Yes, sir. Yes. Good evening, one and all. I am Himani Dabale, and as a convener, I welcome you all on the behalf of Microbiologists Society India. Today, we are going to learn about one of the most interesting topics probiotics and postbiotics in colon cancer. I welcome the speaker for today's session, Dr. Pranav Tambe, sir, Assistant Professor and in charge, ASR Laboratory, Department of Biotechnology, MCE Societies, Abidar Inamdar College of Arts, Science and Commerce, Pune, Maharashtra, India. Dr. Tambe sir completed bachelor's in microbiology from Garware College in 2007 and his master's in 2009 from Ferguson College. Dr. Tambe sir recently completed his PhD in microbiology from Bharati Vidyapit, deemed to be University Pune. Without further ado, let me welcome our speaker for today's event, Dr. Pranav Tambe sir, to proceed with the session. Thank you.
Thank you, Imani. Before beginning, I would like to share my screen. So just please let me know whether my screen is visible or not.
Yes, sir.
Is the screen visible?
Yes, sir. Yes.
Okay. So with due respect for all my teachers present over here, I would like to start my today's lecture, that is on probiotics and postbiotics in colon cancer. Basically, this work was entitled for my PhD, and my entire research, it was performed under the research guidance of Dr. G.V. Madi. So, before I begin, so introduction, probiotics are nothing but these are defined as live microorganisms which, when administered in adequate amount, they confer health benefits to the host. This definition, as per literature survey, was given by WHO in the year 2002, and recently till 2025 there is no change in this definition. So some of the beneficial effects of probiotic consumption include improvement of intestinal health by regulation of microbiota, and also it plays a significant role in stimulation and development of immune system, synthesizing and enhancing the bioavailability of nutrients, reducing symptoms of lactose intolerance, and reducing the risk of certain other disease. Now, these are the few branches or few applications of probiotics. Nowadays, probiotics and postbiotics are widely explored in colorectal cancer, as well as in combating multiple drug resistant bacteria. Also, cholesterol reduction is there, and immunomodulation, lactose metabolism, and food digestion, and diarrhea is also one of the application of probiotic. So, to begin with, how I came to this topic, it was that my interest lies in cancer research. So this slide, it typically shows how healthy colon, it develops into a colon cancer. So in the beginning, when the normal epithelial cells are there, there is a loss of tumor suppressor gene, APC. APC is nothing but a tumor suppressor gene. It is referred to as a gatekeeper gene, which is very important in regulation of beta catenin and Wnt signaling pathway. As soon as there is a loss of this tumor suppressor gene APC, there is beginning or small benign growth of a polyp. In turn, after promotion from 10 to 20 years, there is activation of KRAS oncogene. This KRAS oncogene plays a very major role in signaling, and when mutated, it causes cancer, which leads to loss of tumor suppressor gene DCC. DCC plays a role in cell growth and development, and it helps in promotion of apoptosis. And the major loss of tumor suppressor gene is P53. P53 gene is a master gene, which in certain cancer, either it is upregulated or it is downregulated. loss of P53, but obvious it will lead into development of malignant tumor carcinoma, which will further develop into colon cancer. The mechanisms which are underlying or which are highlighted in the literature for role of probiotics in regulation of colorectal cancer, it include intestinal mucosal barrier, inhibiting pathogenic bacteria, and intestinal cell proliferation and apoptosis. So my interest basically focused on how these probiotics and postbiotics are affecting or controlling the cancer cells, or how there is induction of apoptosis either by induction of Bach-Spuma genes, caspase-3 and P53 genes. So the hypothesis of my title was from relevant literature survey. It was proposed that the isolated organisms, so I isolated certain probiotic organisms from curd samples, and they may play a crucial role in inhibiting and proliferation of cancer cells, and they may serve as a potential alternative alone or in combination to cancer therapy with minimum side effects. Now, one of the basic side effects of chemotherapy, we have seen that many people, they suffer from weight loss, there is vomiting, nausea, diarrhea. So can probiotics play a role in inhibiting or minimizing these side effects? Also, it would promise results in curing cancer by modulating various cell pathways, thereby leading to cancer cells towards apoptosis. So when I started my work, the isolation and identification part of probable probiotic bacteria, curd samples were taken. These curd samples were homemade curd samples, and they were just spread on. Isolation was done by using sterile MRS agar plates, and biochemical tests were performed. So the strategy which was used to fulfill the proposed objectives in my thesis were: first, isolation and characterization of probable organisms was done. Colony and cultural characteristics was done. Gram staining motility, endospore staining, gram positive motile rods, which were typically non-spore formers. They were selected for biochemical characterization, and various biochemical tests were performed. So this is the methodology which I went. 11 homemade curd samples were collected from different sources. They were brought to lab. Serial dilution of these samples was done in sterile phosphate buffer saline up to 10 to the power minus 4. Then these diluted samples were spread on sterile MRS agar plates. The plates were incubated at 37 degrees Celsius for 24 to 48 hours under microaerophilic conditions in an airtight desiccator. After incubation from these 11 samples, I got 40 distinct colonies were selected based on their characteristics, and all these organisms were identified as per the guidelines which were provided by Burgess Manual of Systemic Bacteriology. So these were the sample plates of purified colonies which were used. Now the screening of isolates: firstly, gram staining and motility of the test culture was done. Endospore staining was done to check the presence of endospores, as in literature survey I came across that various probiotic organisms or number of probiotic organisms are typically non-spore formers. So endospore staining was done. So in this, the isolates which I got, LC02, lactobacilli from curd, it was labeled like that: LC08, LC13, 17, 18, 19, 25, 27, 30, 35, 36, and 37. So out of which LC8, LC18, LC20, 25, 27, 30, and 35 were further selected. So the biochemical tests were done. In these biochemical tests, INVIC tests were performed, nitrate reduction was done, hydrogen peroxide production testing was done, catalase test, amylase test, and biochemical analysis of these cultures were done. Catalase test. Now this test was very important criteria for selection of the organisms further, because the lactobacilli group, they are usually facultative anaerobes. So as they are facultatively anaerobic, they do not require oxygen for their respiration, and they don't produce the enzyme catalase. Hence, as they are not producing the enzyme catalase, the organism selected LC8, LC20, LC27, and LC30, these were catalase negative and were further selected for the characteristics. So the results of INVIC test are summarized in this table: indole, methyl red, VP, and citrate utilization. These are the images which I got. So So results of biochemical test: nitrate reduction test, gelatin liquefaction, urease, and starch hydrolysis. Starch hydrolysis was one of the important criteria, as all my 4 isolates, which were catalase negative and non-spore formers, they showed starch hydrolysis positive. Now, one of the criteria for considering the test positive was when the probiotic organisms, they colonize. When they are colonizes, there is extra requirement of carbon. So these organisms, they produce some extracellular enzymes. These extracellular enzymes, they can or they degrade the complex polysaccharides or sugars to obtain their carbon source. So all my 4 isolates, they showed starch hydrolysis, indicating that they may be producing some extracellular enzymes which were causing degradation of this polymer. So sugar fermentation test was done. All the organisms were showing acid and gas production. So the conclusion of biochemical analysis was that according to Berge's Manual of Systemic Bacteriology, second edition, volume 3, the identified isolates LC8, 20, 27, and 30 were categorized up to genus level. The organisms were showing similarity with genus Lactobacilli and were selected for further probiotic properties and their safety properties as per the guidelines which were given for by WHO. So now these isolates were further subjected to different tests for determination of probable probiotic characters, which include acid tolerance, bile tolerance. These 2 tests, they play a very major role in segregating or in identifying the probiotic organisms from other organisms. Temperature tolerance, NaCl concentration, tolerance to phenol, and different enzyme tolerances were checked. Now, acid tolerance. Acid tolerance is one of the important parameter for identifying or for claiming a particular organism as a probiotic organism. Why? Because these probiotic organisms, it is a very crucial survival trait to survive in acidic as well as in alkaline environment, also which is provided in the gastrointestinal tract. Now, how these mechanisms are modified? These mechanisms are modified by the organisms either by internal pH regulation. They may be producing some acid resistant proteins and increased generation of ATP. These are the 3 probable mechanisms with which the probiotic organisms may show tolerance to acid. So a range of pH was selected from pH 2 to pH 11. Here at maximum pH, minimum pH 7, all the organisms showed good tolerance, whereas the organisms were showing tolerance at acidic as well as alkaline conditions also. Next test was performed was bile tolerance test. In stomach, there are bile acids and bile salts which are produced. To survive, they should show tolerance. So the main mechanisms which are shown by these probiotic organisms for bile tolerance is either by activation of efflux pumps, by production of certain bile salt hydrolyzed enzymes, or by modification of various cellular components. So the range for bile tolerance was from 0.1 to 2%, where all the organisms showed good tolerance from 0.1 to 2%. Then temperature tolerance was seen. Literature survey, it showed that the organisms, probiotic organisms, are active against ambient temperatures or at room temperatures. So a variety of range of temperatures were studied from 4 degrees Celsius to 45 degrees Celsius. Good tolerance was observed up to pH or up to temperature, up to temperature 37 degrees Celsius, but drastic decrease in growth was observed at 45 degrees Celsius. Salt tolerance was also checked. Now, why salt tolerance was checked? Because high salt concentration, because the stomach, it is having acidic environment, HCl is produced, and some salts are also produced. In order to combat this, salt tolerance test was done. So it was observed that the organisms were showing good tolerance up to 6%, whereas decrease in salt tolerance was seen from 7 to 10%. So to survive in a stomach and to colonize in stomach intestine, this salt tolerance parameter was very important. The next test performed was phenol tolerance. As we all know that one of the byproduct of metabolism is phenol, and phenol itself, it will act or it will behave like a bacteriostatic or it will stop the growth. It will slow down the growth of these probiotic organisms, which may cause an effect, or which may not allow this probiotic organism to colonize in the stomach or in the intestine or GI tract. So the phenol tolerance was seen from 0.1 to 6%, and the organism showed good tolerance up to 0.5, whereas the tolerance started decreasing from 0.6% phenol onwards. Now this antibiotic sensitivity profiling was done to check whether these organisms are sensitive to specific antibiotics or not, which we use, standard antibiotics which were used, to check whether these organisms are gaining resistance towards the antibiotic or not, to check whether there is any probable mechanism of horizontal gene transfer. Out of the 4 isolate LC8 and LC30 showed resistance against chloramphenicol. Now all these tests which were performed, these were performed as per CLSI guidelines, and the document MO4 and M100 was used for this. So the results were compared as per this sensitivity pattern, and the resistant pattern were compared as per the guidelines which were provided by the CLSI. So these were the results which were obtained. Next test performed was antagonistic activities. Now for this, the test cultures were procured from NCL. Standard test cultures were used. Same CLSI guidelines were followed for doing this test. And all these organisms, LC8, LC20, 27 and 30, they showed good antagonistic effect against the test organisms. So now as the organisms were tested for safety profiling and for their safety profiling, digestive enzyme tolerance was also seen. Now various enzymes like lysozyme, then gastrogene,
तू सांग आता तू तुझ्या कारलेलं उदाहरण म्हणतो तुझ्या वैयक्तिक उदाहरण.
lipase tolerance,
आज संदीपला मस्त वाटतंय
pepsin tolerance, trypsin tolerance, all these tolerance were checked at 50 ppm and 100 ppm, and all the organisms showed very good tolerance at both 50 ppm and 100 ppm.
तुम्ही
Now, surface hydrophobicity is very important
तर मग सगळं
parameter
वाटतं
to
इथं.
check whether the organisms are properly colonizing in the intestine or not. So more the surface or percent hydrophobicity, good are the organisms which can colonize properly in the lumen or in the intestine. So this surface hydrophobicity test was done in order to check the adhesion of these organisms to intestinal lining, survival of them in the gut, autoaggregation, which is very important parameter to be checked to be claimed for the organism to be probiotic. So in which here I use Bacillus clozae. Bacillus clozae is nothing but it is a standard probiotic strain which is available in the market. Now this organism was selected. Why? Because it is used in inflammation. It reduces inflammation. It is also used in diarrhea. So I compared all my activities of these isolates along the anti-cancer activities of the organisms with Bacillus clozae. So the organism showed good percentage of surface hydrophobicity, out of which LC20, LC30 and Bacillus clozae was showing surface hydrophobicity up to 50%. So these organisms were further taken. So these are the results which were taken. Then hemolysis was seen. As we all know that hemolytic properties of the isolates to be checked. Hemolysis is alpha hemolysis, beta hemolysis and gamma hemolysis. Alpha hemolysis should not be shown. Even beta hemolysis should not be shown. So all the organisms LC8, LC20, 27, 30 along with Bacillus clozae, here the positive and negative control used was Staph aureus and E. coli. So all my isolates along with standard culture where comparative analysis was to be done for anti-cancer activity, they all showed gamma hemolysis. So after that, all these LC8, 20, 27 and 30 along with Bacillus clozae, they were inoculated in sterile broth for extracellular protein isolation and estimation. As the probiotic organisms are known to produce extracellular proteins, all these cultures were incubated at 37 degrees Celsius for 4 days. after which ammonium sulfate precipitation using 40% ammonium sulfate was done. The extracted protein was subjected to dialysis, after which partially purified protein was subjected to Follen-Lowry method, and the protein was estimated by using standard graph. The protein concentration which I got was estimated in milligrams per ml. So LC8 yielded 6.9, LC20 yielded 6.2, LC27 5.9, LC30 1.3, Bacillus clausii yielded 3.6. All these protein concentrations were marked in milligram per ml. So before beginning of anti-cancer activity, antioxidant activity was performed. Due to certain metabolism, free radicals are formed, and there is generation of reactive oxygen species. To neutralize these substances, few biological molecules are formed. So my isolates were checked against standard ascorbic acid. Here ascorbic acid was used, here DPPH assay was used, and ascorbic acid is used as standard because it scavenges DPPH easily by donating electrons and decreasing the DPPH absorbance. So all my isolates as compared to control ascorbic acid, they show good percentage of scavenging activity. Now I started my study with two cancer cell lines. One was HCT116 and other was COLO205 cell lines. Now there were various questions. I analyzed almost 8 cancer cell lines, out of which HCT116 and COLO205. HCT116 is KRAS oncogene mutated. The genetic stability of both HCT116 and COLO205 was found out to be more. Both these cell lines, HCT116 and COLO205, were P53 mutant. Because of these two cell lines were selected for my further studies. So the MTT assay was performed on these two cell lines where IC50 values were calculated. So the images are showing the cells before seeding. So in MTT assay is the assay which determines the live mitochondrial oxidoreductase activity. So in this MTT assay is nothing but it is a dye which is yellow in color. So if the organisms or if the eukaryotic cells are having active mitochondria, it produces an enzyme oxidoreductase. This oxidoreductase enzyme converts this yellow colored dye into purple color Forzamin crystals, and these Forzamin crystals are dissolved in tissue culture grade DMSO, where they are estimated and absorbance is taken and the IC50 values were calculated. So these are the plates where 96 titer plates were used and the percent viability of cells was determined. IC50 values were calculated by using graphite prism. Here, out of the 4 isolates and along with Bacillus clausii, standard drug used was doxorubicin, which is also known as ADR. Isolate LC20, LC30 and Bacillus clausii, they showed good IC50 values against HCT116 cells. Here, as compared to Bacillus clausii and Adiramycin, ADR, doxorubicin, LC20 and LC30 showed very good anti-proliferative activities, whereas the same drug on COLO205 had no significant changes. Even Bacillus clausii, it showed no significant changes or no significant IC50 values against COLO205 cells. Now the anti-proliferative activities against HCT116 cell line. The cell extracts LC20 and 30, they displayed potent inhibition with IC50 values were 5.9 and 19.84 micrograms, whereas the standard was showing approximately 27, or not approximately, the IC50 value was 27.8. LC8 showed IC50 values more than 100, which clearly disqualifies the candidate as a potential anti-cancer agent, or we can say can be exploited as an anti-cancer alternative. LC27 was showing moderate activity as compared to Bacillus clausii. So from these experiments, it was clear that the crude extracts were showing good activity, anti-proliferative activity against HCT116 cells, whereas total negative effects were seen on COLO205 cell line. All the cell-free extracts were showing IC50 values more than 200, which is not acceptable at all. As compared to standard was showing IC50 values along 32.6 micrograms. So the experiments of crude extract, we are having concluded that the experiments were showing, or the extracts were showing low antiproliferative or no antiproliferative activities against Holo 205 cells. Now, as the organisms or not organisms, as the cell-free extracts were showing good IC50 values against IC50, it becomes a very crucial parameter to check whether these extracts are having no activities against PBMCs, which are our normal blood cells. So the normal blood cells were isolated and the same experiment of MTT assay was carried and the IC50 values were calculated for all the 4 isolates along with Bacillus clausii and ADR. So all these organisms showed IC50 values, or the extracts or the postbiotics which were obtained from these organisms showed very less effect on the, or showed actually no negligible effect on the blood cells. After that, one of the important aspect of my research was I studied or I performed in vitro wound healing assay. In vitro wound healing assay is nothing but it studies the cell migration and cell to cell signaling. Now why? Because this becomes very important for a phenomenon for cancer metastasis. So here the experiment performed that the cells are seeded in a 6-well plate. Confluency is attained, round about 100% confluent cells are attained, and here the IC50 values which I got for LC20 and 30 in time and dose dependent kinetics manner, LC20 and LC30 half IC50 and double IC50 values were used for in vitro wound healing assays. So for performing this, the cells were seeded after 24 hours of incubation. When the growth of the cells was 100%, the cells were given a scratch by 0.2 mm tip, and the cells were treated with the half IC50 and double IC50 doses. The images were taken at 12 and 24 hours. If we see that the half IC50 values, they behave same as that of the control. Here I have written in the slide where I have mentioned as scratch. The wound which was given to the cells, there was good cell migration and cell to cell signaling for the wells where half IC50 concentration was used. These cells were behaving same as that of the control, whereas the cells which were treated with double IC50 values, these double IC50 values, they signified that there was decrease in cell cycles or cell to cell signaling, and it was preventing or it was slowing down the cancer metastasis. So this led to the further investigation for colony formation assay. Colony formation assay again determines or checks whether the cells are signaling and forming colonies and proliferating or not. Again, for this, half IC50 and double 50 IC50 values were checked. Here, LC20 and LC30 were used. So the results which I got over here the wells which were showing or the different purple color, the entire group, these are the control wells. The wells which I am showing over here, uppercase, these are the wells which are showing half IC50 value treated wells for HCT116 cell line, and the lower wells are showing double IC50 where the cell to cell signaling or migration was stopped and there was no cell signaling. So the cells, they were not confluent as compared to control and half IC50 values. So to understand the mechanistic approach, cell signal or cell signaling, cell sorting was done and cell cycle analysis was done. So it was observed that here also same mechanism was used. Half IC50 and double IC50 values were taken and the treatment was given for 12 to 24 hours. So the cells were harvested at 12 hours also, and the cells were harvested at 24 hours also. So after doing that, there was no significant changes for half IC50 values at 12 and 24 hours. But at double IC50 at 12 and at 24 hours, subsequent population of sub G0 was found out to be increased. So as this sub G0 population was found out to be increased, it may be an indication that these postbiotics or the crude extract which were given to the HCT116 cells, they are slowing down the cell cycle and maybe they are arresting the cell cycle at sub G0 phase. Now there are various mechanisms by which this can happen. Either there is production of cadherins, there is activation of caspases, or there is activation of certain proteins which will induce apoptosis of these cells. So this cell cycle analysis clearly indicated, or the anti-cancer activities, they clearly indicated that the cell-free extracts show good anti-proliferative activities. There was no insignificant effect on normal blood cells, showing less toxicity, so that these cells can be, or the extracts can be used as an potent anti-cancer agents. Wound healing assay showed that double IC50 concentration inhibited cell signaling. Colony formation assay proved the same result, thereby providing strong findings, IC50 findings. And cell cycle analysis, which was done by propydium iodide staining, showed cell arrest at G0 phase, indicating that the cell inclination towards apoptosis. So based on these studies, LC20 and LC30 were found out to be more potent anti-cancer, or the products, or the postbiotics which were produced by LC20 and 30 showed to be producing more potent anti-proliferative activities. So their 16S rDNA sequencing was done, and LC20 was found out to be Enterococcus faecium, whereas LC30 was found out to be Lentilactobacillus paravocenae. So in addition to my objectives which were proposed in PhD, I performed certain docking studies. Now these docking studies were done with thymine 1. So specific proteins which are secreted, or which are found in Enterobacter faecium, that is SAG2A, 2EC8. This is a secreted antigen by the organism, whereas TH1 and other parameters, or other proteins which were checked, were produced, or these are the cell line receptors, or the cell line proteins which are produced on the surface of the cells. So when we perform docking, so it is having certain columns like rank. Rank deals with the order in which different probable or possible binding of ligand within a protein binding site is taking place. Docking score is nothing but it is the numerical value that represents the predicted binding affinity. Confidence score, it indicates the reliability of how ligand fits into the particular protein. Ligand root mean square deviation shows the coordinates of ligand molecules into different structures, whereas interference residues will refer to specific amino acids of protein that are direct in contact with each other. So thymine 1, CD197, CD71, CD95 and CD184 antigens, or not antigens, or the surface proteins which are produced by HCT116 cells were studied against secreted antigens SAG-A to ECA. So the conclusion, the receptor ligand interface indicated that specific amino acid residues, they played a crucial role in mediating the binding interaction between SAG-A peptide and the cancer cell receptors. Structural examination and molecular docking simulations revealed that the amino acids from 409 to 420 SAG-A protein actively participate in direct interaction with the test receptors, and these specific receptors were found in the amino acid residues 71 to 100 and in the regions of 121 to 177. These residues likely contribute to binding through a combination of hydrogen binding, interaction, electrostatic attraction, and van der Waals forces. All binding affinities absorb the docking scores, indicating that SAG-A may modulate cancer-related signaling pathways through its interaction with receptors CD197 and CD71. So these were the conclusions for molecular docking. So in short, summary and conclusion. In my study or in my PhD work, the isolation and characterization of lactic acid bacteria were obtained from different curd samples. The study initially focused on identifying the lactic acid bacteria strains from curd samples, which were sourced from different locations, and as per the guidelines which were provided by Burgess Manual of Systemic Bacteriology, classification was done. pH tolerance, these organisms by initial screening, they were subjected to the safety profiling, and as well as the profiling for probable probiotic characters. And 16S rRNA sequencing showed that the organism belonged to Enterococcus and Lentilactobacillus species. Our findings, they suggested that all the 4 isolates, out of which LC20 and LC30, that Lentilactobacillus paravocenae showed good anti-proliferative activities against HCT116 cells, whereas no significant activities were seen on Colo205 cells. So these are the future prospects of my study, or I'll see that whether in vitro testing, which we have done during my PhD work, can this be done on various animal models and to continue the molecular docking part more, to specifically understand how these crude extracts are combining or binding to the cancer cell receptors or cancer cell proteins, and are they activating certain pathways which are inducing the cells towards the apoptosis. So my entire work, it included 263 references, out of which few I have cited over here. Thank you.
Hello.
Thank you, sir. Yes, any questions that you might have are welcome now.
Hello.
Yes.
Can I make few comments?
Yes, sir. Sure.
Very first, I'd like to congratulate you on very good work and, of course, nice presentation.
Thank you, sir.
The area you worked, some of these are of interest to me. So I made a point to attend this presentation. Can I make few comments?
Yes, sir. Sure, sir.
Yeah. Very first, one of the isolate was enterococcus.
Yes, sir.
Enterococcus, its role in as a probiotic is questioned many times over last 2-3 decades.
Yes,
And
sir.
some of the strains have now been identified as Vizela. But
Yes, sir.
since you did 16S rRNA,
Yes, sir.
definitely these were not Vizela
Yes,
species.
sir.
Right. So that was one comment I wanted to make. Another is you checked for their bile tolerance. Okay.
हाँ, sir.
Hello.
Yes,
You
sir.
check
बोला.
for their bile tolerance, and it looks like the presence of bile is supportive for the growth rather than tolerance or inhibition or so. Particularly at 1 and 2 gram per cent concentration, there was more
Yes,
growth
sir.
at lesser concentrations.
Yes,
Okay.
sir.
One more comment I'd like to make is most of the graphs there, you plotted on y-axis optical density.
Yes, sir.
At somewhere at the very beginning of the experiments, did you correlate the optical density with colony forming units? I
Actually,
think that is
sir, these all my assays which I performed, these were qualitative and quantitative. So I
agreed.
took the OD at 630 and I directly plotted the graph. I didn't measure CFU.
Agreed. But
Okay.
at some stage later on,
Yes,
when you
sir.
go for application point of view,
Yes,
then
sir.
you should know how many organisms remain alive after
Okay.
all its passage through GIT and reach to the site where they colonize. Right.
Sure, sir. I will see.
Yeah.
I will see,
Yeah.
sir.
Yeah. Then another is did you check the adherence to certain cells, colon intestinal cells, adherence of these organisms?
Huh.
Sometimes they use CACO2 cell line
Huh.
to check this. So did you do that, or did you
Caco2 cell
consider
line,
that?
sir. Actually, adherence, Caco2 cell, no, sir. Surface adherence was not seen. I didn't did this.
Okay, because again, going for the application
Yes,
point
sir.
of view, these organisms should not pass. Some of these should adhere to the intestinal cell-like
Yes, sir.
cells
Sure, sure.
and then colonize and then increase in number or so.
Okay.
Right. So you may consider that in your future prospects.
Yes, sir.
Uh, and one last one, I'd like to ask you. You said cell extracts were tested in different cell lines.
Yes, sir.
Right. Did you characterize this extract?
No, sir. No, sir. Characterization, I just gave the samples to university for GC and HPLC.
Okay.
So depending upon the peaks, they said that the peaks which will come, so we can identify. So for
Mhm.
segregation also, they said that chromatographic techniques would be required and it will take a prolonged period of time. So I just studied the crude extract. I didn't went for the purification and determining what were the actual extracts which were present.
Yeah. So that will be your future work.
Yes, sir.
Okay. And then okay, one really last one. Did you
Yes,
check
sir.
for EPS production?
No, sir. No.
Oh, okay. because that will also give some idea about its prebiotic character of these
Okay, sir.
probiotics. Yeah. Anyway, very nice work. You must have some impressive publications as well.
Yes, sir.
And all the best for future
Thank you, sir.
career in this
Thank you,
area.
sir, for attending, sir. Thank you.
Yeah, I had to attend because more than one area was of interest to me. You know that.
Yes, sir.
Yeah. So thank you, Pranav.
Yes,
And
sir.
thank you, Dr.
Yes.
Pranav, and
Yes, yes, sir.
congratulations once again.
Thank you, sir.
Yeah.
Yeah.
Okay, that's all from me.
Okay, thank you. I will interfere only for 1 minute.
Yes, sir.
Really, the presentation of the Pranav was
Pranav, can you hear
excellent.
me?
Yes, sir.
Yes, sir. Only 1 minute.
Sir.
Only 1 minute you give me. Hello.
Yes, sir.
So I want to tell all attendee that anyone who will deliver a lecture on our platform, we are making an appeal now. That is a PhD student, specifically fresh PhD student. We welcome on this platform, and we'll declare very soon that we are giving the certificate of presentation and one memento from microbiologist to society to every speaker. So I make an appeal to all others who are attending. You tell to your friend and encourage more and more PhD students. Thank you.
Himani madam.
Yes, sir.
Hello. There are
Yes,
some
sir.
questions on the
Yes.
comment box.
Group chat. Yes, sir. Yes, sir.
Please ask those questions. Clarify.
Yes, sir. Pranav, sir, the first question we have is what was basis of selecting LC8, 20, 27, and 30 cultures for this study?
Actually, these cultures were selected based on, firstly, I did gram staining and motility. So it was a general selection of isolates, general primary screening of the organisms. These were gram positive short and long rods, and the endospore staining was done. Basically, all these isolates, they showed good tolerance towards pH, bile tolerance, different acid tolerance, bile tolerance. So these organisms were further studied for anti-cancer activity. So I assumed that they may be probable probiotics, and then after performing anti-cancer activity, I went for their sequencing.
The second question we have is from Dr. Narmata ma'am. Did you identify the isolates by molecular characterization?
No, ma'am.
Okay. Third one is from Dr. Prakash Halami, sir. What are specific study needed to select best potential probiotic for CRC?
Sorry to interrupt, but
Yes,
the
sir.
last question, did you molecular characterization? You did that, Pranav, isn't it? 16SR and everything. That is
Sequencing
how
was
you,
done, sir, but
yeah.
sequencing
What?
was done, sir, but actually during my work also there was or the comments from the referees were also why
Mhm.
phylogenetic tree was not determined. So the further part was not done. As phylogenetic trees and everything, they give the evolutionary relationship. I just wanted to know about the organisms up to their genus level. So I didn't went further. I just did sequencing and no further molecular characterization was done.
But that is how you identified the organism, 16SRRNA,
I
isn't it?
identified, sir. I
Yeah,
identified, but
yeah.
their
Without building,
evolutionary history was not determined, sir. So I just know the genus and the species of that particular organism. That's it.
but without building phylogenetic tree, you cannot reach to the species. You find out the percent similarity, isn't
Okay,
it?
sir. Yes, sir.
Yeah, please check with how these were identified.
I outsourced it. I will
Yeah,
just cross verify it, sir.
maybe they did not provide you the phylogenetic tree.
No,
It
sir.
has to be. It has
I
to
just
be
asked
there.
them about the phylogenetic tree.
Mhm.
That time I don't know. त्यांनी एवढंच दिलं सर मला म्हणजे बाकीचं काही ते बोललेच नाही.
No, you check again with that because
Yes,
without
sir.
that
Sure.
you cannot
I will,
find
sir.
the percent similarity to the
Yes.
species and the genus.
Yes, sir. Sure, sir.
Yeah.
I will go through it, sir. Yeah.
Yes. Yes.
Yes.
Okay. Please go ahead.
Yes.
Yes, sir. The third question is from Dr. Prakash Alami, sir. What are specific studies needed to select best potential probiotics for CRC?
What are the specific study needed to select the best potential postbiotics? Now
Probiotics.
typically, yes. So if the postbiotics are showing good anti-cancer activity, short chain fatty acids, example butyrates, lactic acid, where research is going on, there are a lot of research is going on, a lot of papers available in recent editions where polysaccharides which are obtained from these lactobacilli or from probiotic or postbiotic organism or postbiotics, they induce apoptosis. The butyrate which I mentioned, short chain fatty acid, they inhibit cancer cell growth and either they promote apoptosis or the cell death is seen.
Okay. The next question is what type of postbiotics are responsible for anti-cancer activity?
Same. There are short chain fatty acids, polysaccharides, and butyrates, lactic acid.
Okay. Sir, could you please define postbiotics?
Postbiotics are they are the preparations of products from microorganisms or they are components that offer health benefits to the host. They may contain certain metabolites also which may be beneficial to the host.
Okay. Which tool you use for docking?
Autodoc.
Okay. And the last question is, sir, can you differentiate between prebiotics and postbiotics?
Now prebiotics, postbiotics are the what we say. These are the like I defined. Postbiotics are nothing but postbiotics are defined as the preparations or the products from the microorganisms or the components which we are getting. And in terms of prebiotics, we can say that prebiotics are the compounds which are present in the food that foster the growth or activity or they will prove beneficial to the organisms.
Any more questions from participants?
Pranav, can you hear me?
Yes, sir.
Wonderful. No, this is Prakash Alam from CFTRI Mysore.
Yes,
So
sir.
you are one of this culture. What you told, no, that it is a rod, but you are telling it is a enterococci. So that is unlikely actually, because
Yes, sir.
enterococci are focus. They are not the rod, right?
Okay.
Another thing was, you could have selected one of the culture and characterized that one in detail with reference to its colorectal cancer property,
Okay, sir.
which is one of the best among the four which you have selected.
Sir, can you please repeat, sir? I'll just note down, sir.
No, I was thinking rather than you taking a four cultures and struggling all
Yes,
the
sir.
way, you could have selected one of the culture which is a potent and done the in detail study so that what is the postbiotic molecule is, nature of that postbiotic, how potent is that against the colorectal cancer. This is
Means,
what
sir,
actually I was thinking.
I needed to do the I need to separate the crude extracts to find out the components and then check their activity on.
Exactly.
Okay, sir.
Exactly.
Sir,
Right.
actually, this project or this work which you now suggested, sir, this I am planning to continue as one of the cell from the cell line HCT116. Also, I want to check these activities against different cell lines also, like breast cancer and other cell lines also.
Yes. The other thing was, see, yours are the lactic acid bacteria, but then you are comparing with the Bacillus clausii, So they are unlikely that you will get the similar results. Either Bacillus will have better
The
than the
rational.
lactic.
Yes,
So I
sir.
would have suggested that you should actually use one of these lactic, like Lactobacillus amnosus GG or Lactobacillus casei shirota strain, right? You could have
Yes, sir.
taken among the lactic acid bacteria rather than going for the Bacillus. What is your opinion?
Actually, sir, Bacillus clausii, it was a standard probiotic enterogermin which we use. It shows high survival through harsh gut environments and resistant to
Yeah,
various
because that is because
antibiotics.
of the Yeah, that is because of the spores formation.
Yes,
Lactic acid
and
bacteria, they don't form the spores.
and in research, sir, I have seen that they are showing certain immunomodulatory effects and potent pathogen inhibition. So as there is immunomodulatory effects, I just thought if they are showing certain immunomodulatory effects, so maybe they must be showing certain anti-cancer activities. So I just selected on this basis.
Yeah, but then it don't give that much actually very catchy reason to compare with the lactic. That is what I am trying to tell. Because
Okay,
bacillus
yes,
will go with reference to the pH, temperature tolerance because of this correlation nature.
yes, sir.
Right?
Yeah,
And the other one of this audience actually has raised with reference to the enterococci, using the enterococci, because of their very virulency. The problem associated with their virulency. So we usually minimize to use the enterococci as a probiotics because they actually acquire the antibiotic resistance as well as pathogenicity.
okay.
Right. So just ensure that when you are taking for further studies, that
Yes, sir.
enterococci are not selected. Right.
Okay, sir. Okay, sir.
Yeah. Otherwise, they're very wonderful, very good work. You have done the several different studies, and now colorectal cancer actually cases are increasing.
Yes, sir.
So definitely the probiotic therapy is very much required
Yes, sir.
and very needy, because otherwise whatever we drug which we use has a several side effects, and then drug they again actually disturb our gut microbiota also.
Yes, sir.
Where on the other hand, the probiotics actually will be very useful with reference to modulating the gut microbiota as well as making the eubiotic.
Also, sir, along with these probiotic activities, whatever we are getting, uh, if we are getting postbiotics or if we are using this postbiotics in
Okay.
colorectal cancer, we need to also
Yeah.
check the ADME properties of these crude extracts or purified proteins. How the adsorption, excretion, metabolism is taking place. Adsorption,
Yeah. That.
distribution, metabolism, and excretion is taking place. That is
Mm.
also one aspect which we need to study. If
Yes.
we can market these postbiotics or probiotics as a potent anti-cancer
Mm.
alternatives.
Yes. Yes. You use the MRS media only for obtaining the
Yes, sir.
postbiotics?
Yes, sir. Yes, sir.
And what was the so how long you grow the culture? Because it depends on substance excrete in the medium also. So it was the stationary phase culture, right?
Yes, sir.
It was not the log phase culture which we
No,
use
no, sir.
for the purpose.
No, sir. Stationary phase culture.
So how you rate your work with reference to the novelty, or what is the
Novelty, sir.
novelty of your study?
I just wanted to explore how the microorganisms or the extracellular products which are produced by beneficial bacteria,
Very
because
good.
as we discussed, sir, colorectal cancer is an increasing cancer in India, and
Mm.
lot many side effects are seen. So various the chemotherapy, radiation therapy, various therapies are going on. So if
Exactly.
these organisms or the postbiotics, if
Yes.
giving to the cancer patients, if they can reduce the cancer growth, or if they cannot reduce means not to much extent, so in
Mm.
combination with chemotherapy to reduce or to minimize the side effects caused by chemotherapy, can we give
Yes.
postbiotics which will be
Exactly.
healthy,
Very.
which will be
Yes.
healthy in cell to cell? It will also inhibit cell to cell signaling, and it will also modulate some immunomodulatory pathways which will help in controlling cancer. Sure shot cure will not be there, sir, but it will at least reduce the side effects which are caused during the treatment.
Wonderful, wonderful explanation. I was unable to find your publication related to this use of probiotic or postbiotic in colorectal cancer in your Google Scholar actually, because we
Yes,
are
sir.
also working on this. I did not find your publication related to use of
Recently, sir,
probiotic.
the paper, sir, is just online yesterday. It was accepted.
Wonderful,
It will come, sir.
good, good. Congratulations and all the best.
Thank you, sir.
So there is two more questions from
Yes.
participants. Any other cancer cell lines that has not been studied using probiotics?
Any other cancer cell lines mean right now colorectal cancer is selected, but we can test or probiotics like MCF7 is there, which is a breast cancer cell line. MDMB 231 is there. MDMB 2468 is there. RQ is there, and HT29. HT29 is very limited for probiotic research.
Okay. And the last question, sir, can Enterococcus species be selected as probiotic?
Yes, they can be.
Okay. Any more questions from the participants?
Himani, now you can conclude. Huh?
Okay, sir. Yes, sir. Such an incredible lecture, sir. Thank you so much for sharing your research with all of us. It has been such an honor to be a part of this event. On behalf of Microbiologist Society India, I would like to extend my heartfelt gratitude to the speaker, Dr. Pranav Tambe, sir, Assistant Professor and in charge ASR Laboratory, Department of Biotechnology, MCE Societies, Abedar Inamdar College of Arts, Science and Commerce, Pune, Maharashtra, India. Sincere thanks to Dr. A.M. Deshmukh, sir, All India President, Microbiologist Society India, for organizing this event. Finally, a huge thanks to all the participants from all across India. Thank you, everyone.
Thank you, Imani. Thank you, Arvind sir. Thank you, Mani sir, Divane sir, and all the scientists which are present over here for listening me and giving me very valuable suggestions so that I can proceed my research work in a very fruitful direction. Thank you all.
Thank you, sir.
Thank you.